back on track for our final leg of what's been a very, very interesting meeting. And I thank you all for your persistence. Bill Borkowsky from NYU is going to talk to us next about a very interesting study that I'm sure will provoke a lot of comments. Bill?
Rich, thank you for inviting me. In fact, you must have been watching a Monty Python episode, and it's sort of, and now for something completely different. And thanks for remembering that there actually was an interesting study that is not being done, and so it will remind all of us that it was once. So, some of this stuff you've already are aware of, that there are challenges. You've been hearing it all day. You know that simply intensifying heart doesn't work. There have been therapeutic vaccination attempts that have been in and of themselves not successful. And part of the problem, of course, is, as you've heard, that there are reasonable amounts of the virus that are sitting silently, not expressing anything that allows them to be recognized. So what is the immune system going to see? Well, let's see if this works. Page down or enter. Okay. So again, you've heard some of these approaches from the speakers in the morning. Well, okay, virus is sitting quietly. Let's try to wake it up with histone deacetylase inhibitors or demethylating agents. And of course, these are not benign drugs. And as well as, again, you heard this morning, Bob Silicano using a somewhat modified way of looking at what kind of virus comes out of these cells treated this way. It doesn't seem like, currently at least, the compounds we're testing will really work in terms of activating virus. And in terms of therapeutic vaccination, I want to remind you, you heard about the Merck study, is that you're being vaccinated with parts of a virus that are not in your body. And your immune system actually would like to deal with viral antigens that are expressed by your body viruses. And if they're not present in these vaccines, then you're really not going to be able to deal with any activated virus. The other problem is that even if you take out the most recent virus, if you're not talking about a situation where there was an acute infection, where there hasn't been a quasi-species diversification, that there are founder viruses and there are all sorts of quasi-species that evolved over time. And whatever you have at the end in your plasma only represents a small fraction of what's actually sitting in you. Although, again, what I'll remind you, because it becomes relevant, is that when you stop treatment with antiretrovirals, as was done in one of these Croy studies that this year, it was abstract 388, that what comes out is not just the most recent virus, but in fact, lots of founder viruses and many different quasi-species. So potentially, there would be expression of a panoply of viruses that have been living in your body for a while. You also heard that there are monoclonal antibody cocktails that have very high neutralizing activity. But again, what's interesting is if latent virus doesn't express any viral antigens, then what is there to neutralize? And then the other approach is, well, let's mix and match all of these. So 10 years ago, a number of us, Lewis and I, thought that one could use treatment interruption as an autologous vaccine. And let's see what happens. And the idea is that it would increase HIV-specific immunity to your own virus. And as well, a side effect is that you get TAT expression or TAT overexpression, which has a variety of effects. Some people might say bad, but while we're trying to find agents that are reasonably good at reactivating latent DNA, the virus figured it out a long time ago, and TAT is the best reactivator that I'm aware of. So that not only gets expressed in the cell as the latent virus is transcribing and translating, but it also is released into the environment, and you can get measurable levels of TAT in plasma, and certainly reasonably high levels in the immediate environment of the cell that's being reactivated. Now, the other thing is, even if you had a good therapeutic vaccine and you decided to interrupt your antiretroviral treatment, there will be some virus that comes out, and that virus can actually initiate new infections. So trying to reduce the level of latent DNA is not easy if you can't prevent that from happening. So this just really summarizes all of that. I think the audience is sophisticated enough that I don't have to go through this, but the idea is virus comes in through CD4 and CCR5, uncoats, becomes DNA, which integrates, which under proper circumstances with stimulation will ultimately result in more virus and production of TAT, and in the bottom, expression of viral antigens on the surface of the cell. So here's a little snapshot of sort of how I would envision things happening. And when I say I don't want to leave out my colleague Ramyogev here from Northwestern, who's been a partner in crime with me all these years, and also an immunologist, Betsy McFarland from Denver Children's. But the idea is if you stop treatment, you have cells that already have a small amount of virus. There'll be more. There'll be extracellular TAT. There'll be that TAT going into latently infected cells. All right, thank you very much. And again, this will now express antigens. This will express antigens. There'll be cells that will recognize these and potentially kill them. And there'll be virus. But if you can interfere in infecting previously uninfected cells with something that blocks CCR5 and prevents integration, then maybe you won't refeed the latent pool. So here's a study that, again, we initiated over 10 years ago. It was called P1015. It was done in 11 HIV-infected children who had had undetectable virus for over a year. And they were ages between 6 and 16. Now, previous treatment interruption studies were done, as you've heard, but they were done in a sort of rigid way. That is, one study said, let's stop for a week. Another study said, let's stop for 2 weeks. In the SMART study, it's let's stop until you develop falling CD4s and disease. But we decided to be very conservative and to do this sort of tiptoe approach into the ocean of treatment interruption. And so we devised these cycles where you started out, you were on therapy, undetectable, you went off for 3 days, you went back on treatment for up to 4 months to resuppress virus. Once at the first point you were resuppressed with your virus, your viral copy number was under 50, you initiated the next cycle, which was 2 days longer. So we had 3 days off, 5 days off, 7 days off, 9 days off. And the ultimate goal was to actually get to 49 days off. And we had a whole variety of safety measures that were built in, which was CD4 couldn't drop by 25%. You had to be able to resuppress virus within that 3 to 4 month period after the treatment interruption. and you couldn't show that there was a new mutation to the polymerase region that would render your virus less treatable by the antiretrovirals that you had. And we built into this virologic and immunologic assays looking at CD4 and CD8 responses to HIV antigens. And these were done at the end of every interruption. And as we got down into these longer interruptions, 29, 31, 35 days, we introduced an intermediate time point so that we could actually compare the immune responses and the return of viremia at this point in time, which was actually comparable to this point in time. So we had 2 time points. And what... All right. A frozen screen.
Bill, there's his interface.
Asleep. Let me see.
Okay. So I will tell you a couple things. One is that there were no safety issues. There was a review, and they decided that there were no safety issues whatsoever. The other thing I'll tell you is that there were 2 individuals who developed each of them developed 1 new mutation, so they were removed from the study. But interestingly, all of these individuals went back on the treatment that they were on before, and they were still suppressing their viremia. There was 1 patient who didn't get to under 50 at the end of 4 months of treatment, had a value of 75, so had to come off the study, although the exact same specimen run 2 more times showed that it was actually under 30. So we were a little bit too strict in terms of our safety phenomenon. And so in spite of the fact that it was actually deemed safe, some powers that be decided that there was nothing left to be learned from continuing this study. And so the study was stopped early. But there were 5 individuals who had gotten out far enough to 35 days or more that we wanted to look at because we had a significant amount of data. And what I want to show you is, so here's what their viremia was at, in this case, 17 days off meds. And then 10 cycles later, at the same 17 days off meds, 4 out of 5 had what amounted to a 2-log reduction in viremia. And the one that didn't actually started out with very little virus to begin with. When one looked at CD8 responses, these same 4 that had the 2-log drop had essentially a 100-fold increase in their CD8 ELISpot responses to the virus. And the one who actually didn't really move, didn't really have much to begin with, and didn't acquire anymore. So that's the outlier. If you looked at the whole, what was 8 individuals left, here's sort of what happened. First of all, so here was your baseline virus, and with increasing cycles and time off therapy, it basically peaked over here, and then over time, the viral load went down by a little bit over a log. And contemporaneous with that was the development of immune responses, both CD4 here and CD8, to HIV antigens. So at the very time that viremia is going down, immune responses are going up. And believe it or not, I think this is one of the first studies that showed this correlative effect. The other point I want to make is that it took 6 to 9 interruptions before we could actually demonstrate an increase in cell-mediated immunity. Now, was that because the amount of virus here was insufficiently high to trigger that? Or is it similar to what you expect from vaccines, where you need multiple exposures to an antigen to really boost responses? That was one of the things we wanted to find out, but since there was nothing left to be learned, we never learned it. So the results. The peak was short of 30,000, declined to under 10,000 after cycle 10. The CD4%, which is what we measure in pediatrics, really didn't change. The CD8 increased slightly. There was a tenfold or more increase in immune responses in 7 out of 8, and actually in those 4, it was 100. Basically, I think we were doing well. So that study ended. Of course, at the same time, what you heard referred to, the SMART study came out. And the purpose of the SMART study was not to improve immunity. It was to reduce exposure to antiretrovirals and the toxicities that go with them. And as you heard earlier, there was, first of all, the patients were not 6 to 16 years old. I think the median was 43 years of age. In the group that was randomized into stopping treatment, their CD4 counts dropped in many of them, and they had an increase in cardiovascular disease. That study was stopped, and it made treatment interruption a dirty word. And virtually every treatment interruption study stopped at that point. Well, I didn't really think it was a SMART study, and so we decided to create a strategy to modify antiretroviral therapy to eliminate retroviruses, or the SMARTer study. And the objective here was, in some ways, the same: to increase HIV-specific immune responses, to reduce viremia by exposures to homologous virus, but also to address what would happen to the latent reservoir, and of course to maintain safety. The design was more or less the same, and yet not. And this isn't words, but I'm going to show you a picture to make it more digestible. But first of all, this tiptoeing effect took a long time. And did we really have to only go up by 2 days at a time? And we decided that instead of doing that, we would go to 4 days, 7 days, 10 days, 14. Essentially, we cut the length of time by a third in terms of getting to the point where we think we could stimulate appropriate immune responses. We decided to stop at 35 days because actually, even though that study stopped, I had one patient in my site who, through my IRB, I got permission to continue her on, and the parents signed a new consent. So she went out to 49 days, and it wasn't clear to me that going beyond 35 days actually added anything to further increasing immune responses. And that we would also introduce a new twist, which was at some point, once you actually could increase these HIV-specific immune responses, one would, in the treatment interruption, the only thing we would be interrupting was the protease inhibitors, the NNRTI or the RTI, but in its place we would put something that would inhibit CCR5 and CD4. And although originally we wanted these as monoclonals, the companies wouldn't let us use them, so we decided to go with Maraviroc and raltegravir. And so this is what it looks like now. Essentially starts out the same way. Instead of 3, 5, 7, it's 4, 7, 10, 14. We do this until they get to 35. We do the same looking at immune responses and viremia. We do the same looking at midpoints to compare what happens. And after cycle 10, which allows us to get to 35 days off, we then, while stopping the antiretrovirals, we introduced these two drugs in its place, the idea being to, as virus is released, that it hopefully won't refeed the latent pool. The endpoints, again, how does one know what the appropriate endpoints are? But we're looking for, again, a greater than 10-fold increase in HIV-specific immune responses. And we were also looking for what we thought was a significant effect on the latent pool, which is a drop of a log or more in terms of the DNA. And actually, W. Prasad was also going to be looking at actual replication-competent virus. And again, looking at safety. So this concept was introduced 4 years ago, and it's still sitting on someone's desk somewhere. At one point, just to finish, again, anecdotally, I want to say that one of the things Rich mentioned was, okay, you can increase HIV-specific immune responses, but how long will they last once you sort of stop doing this stuff? And so here's the anecdote. I had a brother and a sister who were a year apart from each other, the sister being 1 year older. They were HLA identical. By the way, I should add that none of the patients in the study had any good HLA gene that would have helped them here. They were HLA identical in terms of class 1. The girl went through the treatment interruption. The boy stayed on his antiretrovirals. The girl's peak HIV RNA was 35,000, and at the end of, she's the one who went out to 49 days. She was down to 2,000 at that 49-day point. She and her brother both disappeared from care and returned 18 months later, not having taken a single antiretroviral for those 18 months. The girl's viral load was 2,000. The boy's viral load on 3 occasions was anywhere from 60 to 100,000. So it's an anecdote, but the fact is that that boosting, I think, actually provided ongoing better control of viremia without the benefit of antiretrovirals. Is this just going to eliminate the latent pool? No, but remember, we're not going to find too many newly infected individuals. Most of the people we're talking about, as has been mentioned, are people who have been infected for long periods of time. We'd like for them to have less exposure to antiretrovirals and lower viral loads in the absence of that, and this is one approach.
Thank you, Bill. In the interest of time, I'm going to ask that we save questions until after the next speaker. And I'd like Luis Montaner from the Wilstar Institute in Philadelphia to tell us about another study.
Thank you, Richard, and thanks for holding through the program. Well, I hope to wake you up if you're asleep, and if not, keep you entertained. So, first slide, how we get, let's see. All right, so I figure what I do with my time is really sort of discuss with you sort of where we are with our concept and our original observations with interferon in adults. We've been actually studying treatment interruption, immune recovery for 15 years, and we've actually followed people interrupting therapy upwards, at least 150 people or so. So we've gained a lot of insights into what happens when and relative to time on therapy, what immunological functions are recovered, what we could track relative to the impact of acute viremia, and what may track with set point after you stop therapy. And sort of all these sort of conceptual ideas are sort of channeling into a common strategy that I'm going to share with you. All right. Here we go. So I sort of organized the discussion on 3 different areas. One is just to give you sort of our working hypothesis, the basis for our working hypothesis as to what do we think we're actually tracking with interferon. And that comes from the work we've done leading to the trial in 2013 that we published and what's happened since. I want to tell you about 2 studies that are underway. that are trying to explore this further. And lastly, I'm going to share with you some, for the sake of the workshop, some sort of preliminary data and sort of present some ideas for discussion as to what may be, what I consider is a shift in at least my thinking that was impacted by some preliminary data that we're starting to see. I think just to review what is driving our interest, we had as I mentioned, we've been studying patients under antiretroviral therapy for quite some time, and we sort of posited the question that if we are to gain from immune reconstitution, perhaps type 1 interferon responses will be a lot more powerful if they are exercised within the context of a fully functional immune system. I mean, the presence of interferon as an antiviral is obviously well documented, but its effects on viremia were temporal, meaning that they were both muted, they were like 0.5 log decline, and it was also released shortly thereafter. But this is given an interferon regimen within a person that is otherwise immunocompromised, meaning highly viremic, low CD4, no function. And the more you sort of review the way that interferon may actually have its effect biologically, the more you sort of come to appreciate the networks that it's actually trying to elicit. And from that rationale, we said, okay, if we take our individuals that are fully recovered by heart and we give them interferon and we allow for the immune system to be primed for 5 weeks and the pharmacokinetic levels to be reached for interferon, what will happen if we stop antiretroviral therapy and continue monotherapy thereafter? The immune activation hypothesis would say that we've sort of juiced up the system and there's going to be a crash and burn immediately after we stop antiretroviral therapy. The hypothesis that we were after is that the natural killer cell networks, the intrinsic resistance factors would all sort of come into play, and then all of a sudden we're going to see a much more potent antiviral response than what had been observed to date. So we powered the study on a treatment interruption sequence where we were looking at viral control, because at that point the question is, would a monotherapy achieve control? And that was defined as a viral load less than 400, 12 weeks after an interruption of antiretroviral therapy. So from that perspective, we would have expected only like 4% of our cohort to meet that criteria at random. So that if we assume that into the power calculations, we only needed 20 people to do this experiment, because we then said, well, interferon is very toxic, so we're going to do both the therapeutic dose given for hepatitis C, and we're going to give the drop-down dose, the 90 dose. And then at that point, we were thinking, perhaps the individuals receiving the 90 dose would sustain this therapy longer, because they would not drop out due to toxicity. And so that was the idea. We recruited about 21 individuals, and here they are listed. I just want to highlight that they were highly recovered for CD4. So these were otherwise great candidates of having benefited from antiretroviral therapy. They were randomized for the 2 doses. And the FDA, when we sent this back to them, said, you don't need a control arm because it's well documented that people have rebounded after they stop therapy, so just use your own historical controls of individuals that you have followed in Philadelphia after they stopped therapy. So that's what we did. The strategy, as I mentioned, is illustrated here. You can see that over the period of follow-up, they were given Pegasus at the week visit 8 for 5 weeks, and then they stopped antiretroviral therapy. Primary endpoint was at 12 weeks at week 25. And if they were below 400, we would then allow them to stay another 12 weeks for 24 weeks. Again, trying to mirror the antiretroviral therapy-based designs that look at suppression for 24 weeks. And we will look at both doses. This was our historical control, where we had already documented that people that stop therapy rebound quite efficiently and quite predictably within a short period of time within the context of follow-up. So against this control, we then looked at the rebound rate from our strategy. And what we observed was that about 45% of people were able to sustain suppression below 400 at 3 months. And of those that elected to be out of the strategy were not due to failures. Those that stayed actually stayed for the full 24 months. So this was quite a surprise, because we were not expecting this level of success when nothing else has worked, IL-7, IL-2, therapeutic vaccines, nothing that elicits host control has accounted for a suppression below 400 or below 50. There were 9 individuals that stayed below 400. 6 of these were below 50, of total of 20. Of the 11 failures, 2 of them were removed due to the fact that they were feeling a little bit depressed, even though they were still suppressed. So they were counted as virological failures from the point of view of the primary endpoint. But nevertheless, this to us illustrated the fact that there was an effect of interferon when introduced to an immune reconstituted system that was quite distinct from adding it in the context of viremia. Then we did an ad hoc analysis, which we went across the street to Una Doherty, who had been looking at integrated levels of DNA in the context of CD4, and we asked her to measure the integrated DNA level within PBMC that we gave to her from either the start of the trial when they were randomized to either side, the time that they stopped therapy, antiretroviral therapy being the second point, or the primary endpoint being the third point. Then obviously this is ad hoc secondary, so we knew outcomes. So we gave her a group of failures, people that went above 400 at week 12 or before then, and successes, people that were at below 400 by 3 months after that. And the surprise to us was that we were able to evidence a 3- to 4-fold reduction over that short period of time in the amount of integrated DNA within the CD4 of those individuals that were clinically responding to the lack of viremia in the plasma as measured by a viral load below 400. So we then sort of concluded that this provided the first proof of concept that a host can be modulated to control through interferon alpha-2A immunotherapy, and that it could allow for a reduction of integrated DNA. Now, obviously, the biggest question that we asked, even though we had 9 responders, whereas we were sort of very enthusiastic with one person getting to below 50 in the CCR5 gene modification study, we're very enthused about the individuals that have been able to gain from particular strategies. These are 9. So this is by no means meets a sort of a one-off type of definition. However, the question is, can you do it again? Because maybe these people were, I don't know, really enthusiastic, or whatever reason you may want to come up with as to why we would see 9 individuals suppressed, even after confirming with the pharmacy that they had not refilled their ART relative to, that was one of the first questions the physicians in the study asked us to address. So this month's JID has a report from Harvard actually reporting the similar result, which is a reduction in integrated DNA, but this time, rather than in our study where we looked at mono-infected, HIV mono-infected people, they were looking at the change in the reservoir after the tenure of an anti-HCV therapy inclusive of interferon. And they also describe a decrease in the PBMC at the end that is actually sustained at the SVR time point, which is 24 weeks after the stop of interferon. So this now becomes 12 patients in their study, 20 in ours, of which 9 were responders. Not everyone responds, and we can talk about reasons why that is, but I don't have time to go through everything that has been done in this study. But to give you a sense of what is it that we think is happening, why did it work when others didn't work? And the way that we're thinking about it is that interferon could elicit a highly active antiretroviral response. And of course, I'm taking off from heart by saying that it is really a composite of multiple mechanisms. It's not a one mechanism type of approach, because I think that interferon is well known to induce interferon-mediated gene expression, which goes with intrinsic host factors. We know that it induces cellular innate responses like the natural killer cell response, and there is some evidence that it can also elicit antibody production from B cells, and it's instrumental in actually allowing a naive CD8 response to actually move forward. So within this framework of interferon being reflective of a sort of a general host mobilization of antiviral responses, our study was categorical, was either control or no control. So we then sort of shifted into other cohorts to tell us what the relationship between at least the innate immune response, which we saw was higher in the controllers of our interferon study. And what I mean by that is K562 lysis by PBMC, which we know is a cancer target that is actually lysed by natural killer cells. This was an activity that was greater in those that were responding to interferon clinically. We also know that ApoB and tetherin and all the interferon-mediated genes were upregulated by interferon. But that wasn't enough, because most upregulated interferon, those that did not, were in the non-responder group. So now we have 2 different things that need to be together: the interferon-mediated gene induction and the natural killer cell response. so that the CD8 is still to be measured. We have the samples, and hopefully if the R01 ever gets funded, we will have that answer, because we want to fill the blue spot as well. But I certainly think of it from a comprehensive point of view and not a single mechanism point of view. However, I'll share with you several observations from 3 different cohorts. This one is from the treatment interruption control group, the one that everyone rebounded. If we look at the immune variables at the time that they stopped, and then we actually regress that to the viral set point that was obtained sort of 3 months later, and we say, what was it at set point that correlated with the level of the viral set point taken much later after that? And one of the things that actually came back as inversely related was the amount of plasmacytoid dendritic cells. The reason this is intriguing is that that's the subset that makes interferon alpha, for example. So our strategy was almost like remove the PDC and just give interferon, because the inference is that if PDC were actually contributing to a lower set point without therapy, then you should be able to replicate that function by giving one of its main mechanisms of action, which is interferon. And that's certainly what we interpreted. That occurred in our study. The next is we looked, and this is consistent with other individuals that have reported this relationship as well, in acute infection, I should say. The other was a study in elite controllers. If you're telling me that natural killer cells can suppress, then find me elite controllers that are below 50, functionally cured, if you want to call it that don't have or do have the alleles that track greater natural killer cell function, and show me the balance between their CD8 function and their NK function. At least that was the question. Because if I can show you that there are individuals that are suppressed and have higher NK function without a high CD8, then I would be more comfortable in interpreting that if I move the NK response, I'm going to get clinical suppression. And that's indeed what we reported in this 2012 report in AIDS from the cohort with Stephen Deeks, where we basically tracked the K562 response in the presence of interferon on the y-axis and the GAG-specific interferon gamma response in the CD8 on the x-axis. And we were able to see at least an inverse relationship between the amount of NK and the amount of CD8. Now that this falls apart if you have a viremic individual. These are elite suppressors individuals where there's very little antigenemia coming from the plasma viral load. But it allowed us to interpret that the NK could be identified with control when CD8 function was low. So again, it goes with the overall hypothesis that I was trying to put forward. The next is a study of individuals that are what do we call viremic controllers. They're not elites, they're between 50 and 2,000. They have a CD4 count above 250, and we're trying to determine what is it unique about them when compared to a viremic person that also has a viral load above 250, so they're not really advanced, and they have a set point of about 20 to 30,000. We know that factors like T-cell activation is going to be equal in both, like CD38, because they're both viremic. And we know that they're not advanced because they have high CD4, but we certainly see different viral loads. So what is it about their immunological profiles that we can learn associates with control? And you may or may not be able to see all this. This has been submitted, and again, I don't have time to go through each one of these little statements where I can show you data for, but what I want to highlight is that one of the things that is shared in low viremia and high viremia is obviously the activation of CD8 and the spontaneous degranulation of the NK. So this becomes almost like activation markers of viral load. When things start to diverge is in the frequency of PDC being greater with suppression than not, the frequency of the NK being greater and retained in low viremia than high, the GAG to POL ratio of the CD8 response being greater for GAG in the controllers and greater for POL in the viremics, again because GAG is more conserved than POL, so that if you have a response against GAG, you will control, or at least that's the hypothesis. So then we did a multi-regression analysis and models to try to determine what may predict for the continuous variable being the viral load, because we didn't have that, for example, for our study. We had controllers or non-controllers with interferon. And the outcome of that modeling identified the PDC frequency, again, as a significant predictor of about 52% of the variance of viral load, and the amount of CD8 responses against POL, which again gives you this idea of a balance between the innate and the adaptive in helping to explain what is otherwise a continuous variable of viral load. And the question is, well, what may account for the other 48%? because we can only account for 52% of the variance, but it gives you this idea of innate to adaptive. Well, we would put forward that things that we didn't measure are issues like the interaction between interferon-mediated genes and CD8 function. We know that the more you stimulate factors like TRIM1 alpha, you may actually spill into a differential susceptibility for CD8 control. We also know that if you respond to specific epitopes in the CD8, you may then alter the KIR detection of the NK coming from alleles that otherwise are controllers. So this issue that there is a crosstalk between your NK antigen sensitivity for KIR, your CD8 epitope selection, and your interferon-mediated gene expression that may impact the balance between how these three interact is something that I think we start to see more and more evidence for, but we seldom do it together because we either do an ELISpot for CD8 or an NK activation or a TRIM5 expression. But I think that the idea is that I'm trying to put forward is that we need to think about this in a more global fashion, because I think it becomes a lot more clearer what the correlates of control are. So this is sort of like how I see the world relative to interferon-mediated effect, where we have the interplay between intrinsic, cellular, immune. We have the crosstalk that naturally occurs between your type 1, primarily with intrinsic cellular, certainly emulated by the type 2s in the interferon gamma in the context of a functional innate and intrinsic, which would not be the case in chronic viremia. And this may or may not be exploited by interferon in the absence of chronic activation, which would be in the context of viral load. It's intriguing to me, at least, the fact that the transmission efficiency is very low for HIV, and we know that interferon responses are one of the first things that are part of that exposure. So to that extent, that becomes of interest. We're planning to move forward with this with a pilot in 25 individuals, which already got funded, and it's currently recruiting, and I'll show you the design for that. And we also just received funding for the U01 on 54 patients that will be recruited. So we'll have 79 individuals undergoing interferon-mediated immunotherapy, followed by treatment interruptions to learn whether this data could be built upon in learning more about what is the control. The pilot is here. You can see 20 weeks of interferon. We shifted to interferon alpha to B because it has greater tissue penetrance than interferon alpha to A. And we're recruiting. We're currently at 10. We're recruiting about 2 per month. And you can see that there's a 4-week interruption, again with the idea of awakening the latent reservoir. These are 2 patients we're going to come back to. I just want to highlight the fact that they were diagnosed in 08 and 03. They're in a triplet since 07 and 08. And they were stably suppressed even to the point of last November when they joined this pilot, which is below 20. So they're like, you're good, solid, suppressed. And we'll come back to these folks. This is we call a BEAT study, and this is the design. We now have the funding for a full-fledged randomized trial where we compare nothing to interferon on top of ART for 20 weeks, or the same type of design as the pilot. However, when this went through the NIH, they changed the design, and they asked us to extend the interruption until the virus is detected, again with this idea that we need to wake up the latent reservoir. It's almost like your poor man's reactivation, remove ART. So with that, this is projected to accrue in September, whereas the pilot will end by October. So we'll have data by early next year relative to the outcomes of the pilot, whereas the randomized trial will probably give us data in 16, beginning of 17. And the primary endpoint in both the pilot and this is the change in integrated DNA. It's no longer control, but it's actually the load of HIV within the context of the PBMC. Secondarily, we're also taking leukapheresis at the beginning and at the end. We're taking rectal biopsies at the beginning and at the end because we want to know whether we actually extend suppression to the tissue as well. And that's the part I want to highlight now, because in the context of the latent paradigm, where we have a stable integrated DNA, proviral DNA within the cell, the idea of this is that it's a one ball in a million, and we have to find it, and we have to awaken the virus. And where is it? And therefore, we need HDAC inhibitors. We need to get a treatment interruption in the design, because without that, we won't see the latent reservoir. And I've been intrigued by the now increasing amount of reports that document that persistent HIV in the tissues is a very accessible and documented piece of an antiretroviral treated individual. There was a report by Tim Schacker's group on the lymph node and the presence of HIV RNA in spite of undetectable levels in the blood. There were other reports by the Palmer's group that actually did sequencing of early and chronic and shows that there is some degree of movement in the sequences in the rectum, suggesting that there is evidence of ongoing replication or ongoing persistence in the tissue, even though we can't see it in the blood. Well, remember the two individuals that I told you that we did a baseline assessment of rectal tissue and leukapheresis because they're currently on interferon? Well, when we look at these two folks and we look at the in situ hybridization signal for HIV RNA in the rectal tissue, we sort of sent these tissues over to Q Lee, who's in Nebraska. He came from Ashley Hass's group. And he helped us identify, at least initially, the signal for HIV. And here's where I was surprised to see this, which is that you can see this green is a pseudo color for black for the in situ signal. And what becomes evident is that in the tissue montage, the lymphoid aggregate is not where the green signal is, but the submucosal regions proximal to the epithelium of the mucosa, of the rectum. And these regions are repeated throughout the tissue. And I have a higher magnification. I mean, I basically blew it up, and you can see that the relationship between the lymphoid cluster and these sort of green areas, which we are interpreting as RNA expressing cells. Now, this is PIP2. If we look at PIP3, we get the same type of image, where we get the detection of the RNA in the tissue when it's not in relation to the lymphoid organ. Now, just in March, there was a report that came out on the accumulation of macrophages in the gut relative to a viremic individual. And what I want to highlight to you is they were tracking the ileum, and they basically show that in a viremic individual, there's an engorgement of CD68, 163 cells in the epithelium, and that this amount of an expansion attritions back with therapy once they measure them on antiretroviral therapy. So I think my interpretation is that we're tracking in the tissue the residual expansion of what may have been a macrophage-mediated infection in the tissue that is not evident in the blood. And to that, it brings up the question of what is the balance between blood and tissue, and are we tracking the latent in the blood when there is an excess of persistence in the tissue? Are we tracking the CD4 cell in the blood when there's an excess of myeloid reservoirs in the tissue? And which strategy and by which measure are we going to be evaluating a cure strategy? Because if the tissue persistence is a major component of what we face, then the blood becomes a small fraction of what we need to be monitoring in order to advance a cure strategy. And obviously, it's early days. I just showed you two patients. Both of them happen to be in a tripla, so maybe when other patients come, we'll see whether this is replicated in other individuals. But for me, 2 is enough to give me the confidence to say that it is possible to detect it, in spite of the fact that these individuals were poster childs for a cure-related strategy from a point of view of suppression and immunological recovery. So we'll see if interferon can address tissue expression in addition to the integrated load. So now we have two different things we could be looking for: the amount of RNA expression in the tissue and the amount of integrated DNA, if there's indeed NK-mediated clearance, as we think that we will see. So with that, I mean, the team is actually very large and in relation to these studies, and as these 79 individuals become recruited and followed, I'm sure that there will be more to learn from this. And whether interferon is going to replace antiretroviral therapy, no. But I do think that if we can establish that interferon allows to go beyond antiretroviral therapy, and it shows us what and how to manipulate an immune system towards control, I think that we could either consider interferon in a future combination strategy, because we know it works, at least temporarily. And second, we may also explore how to exploit these pathways with perhaps more targeted interventions than interferon as a whole. So that's kind of like where we're headed. Thank you.
Yeah, thank you very much. I think we're a little behind time. I'm going to invite Dr. Borkowsky to come back and invite questions from the audience. If there are 1 or 2 burning questions for our guest speakers. So it looks like Rich has a question.
Luis, I'm very curious if you've thought about this strategy in recently infected patients instead of in chronically infected patients?
I think that's a really intriguing scenario because here we already know that the initial host resistance factors, whatever they are at the time of exposure, have not been sufficient. So now we have what is otherwise the increase of interferon associated with increase in viremia. And the question is, would early treatment, would we see something like the Visconti cohort with a greater frequency than 15% if we were to introduce interferon at that point as a temporal intervention? It's intriguing. However, I would also expect that the host is committing an interferon response naturally. So therefore, we don't know to what extent is that part of why we see the drop in viral load that we see. So the question would only be relevant if the mechanism is limiting, meaning that we can actually get more than what the host is already doing, because we do see interferon go up.
You have another question?
One for Luis. So I'm Reuben Harris from University of Minnesota. So when you're treating with interferon, did you happen to clone any sequences and look to see if they're hypermutated by APOBEC?
That's a great question. We were just discussing that we have not established that, because we view viral load as, if measured, bad. And it is still possible that due to APOBEC, that viral load in the presence of interferon is not as infectious as in the absence of interferon due to the hypermutation rate. But we haven't done that. We should do that.
So I was intrigued by your in situ hybridization experiments, and was there, I'm just curious about some details of that. So, like, was there one biopsy per person? Was there more than one biopsy?
There were 16 biopsies obtained. 14 of them, because you take two pieces per each pass, is what the gastroenterologist sort of does, with endoscopy. And 2 pieces of this is going into fixative, and those 2 pieces are the ones that have emanated this data. The other 14 pieces are being made into a cell suspension. There's flow cytometry being done on them to determine frequency, to address redistribution, and a cell pellet for RNA and integrated DNA. Because that's what we initially were expecting was the most robust measure, because of what Stephen Dix has reported and others relative to sort of rectal tissue analysis. I certainly did not expect to receive a clear indication of expression in relation to the tissue, because I thought it would be a hit or miss.
Right, but if you sort of extrapolate, so all of your guys had an in situ signal, right? So if you extrapolate that over the surface area of the entire gut, there should be a ton of virus there. There should be gazillions of viruses there.
I agree with you.
So that says to me there's something wrong with that picture.
Okay. Well,
but your PCR will tell you. But if you're looking for something rare and you find it easily, you should be very suspicious.
But if you look at all the reports to date, at least that I have seen, the Stephen Dix, we looked at, he's published about maybe three reports on analysis of rectal versus ileum versus lymph node. If you look at the if you look at the data that has been reported till now, there's a couple of observations that everyone has been reporting. Number one, that RNA is a lot easier to measure than DNA because it's in excess in the rectal biopsy that you obtain.
But that's by PCR, right?
Yes.
Okay, so.
So then
PCR will tell you the truth.
No, I understand.
And then you can call
but what I'm saying is that up to now, there has been nothing to tell us what is the relationship between a signal and the amount in RNA in the PCR reaction versus the detection of low copy numbers when they go above 20 in the plasma.
No, I know, but think about that picture. That's not low anything. That's a ton.
Well, it's actually if you look at the entire tissue, you're talking about 10 sites.
I think we'll not.
I have one question, if you allow me. So plasmacytoid dendritic cells are supposed to disappear with ongoing infection, or so is thought, but a recent paper suggested that it's not that they died, but that they went to the gut to live there instead. So the question is, so you're correlating the higher level in the blood with a more prolonged response, and that it's partially responsible for that, but it may be that it's the reverse. That is that there's less virus around, therefore more plasmacytoid dendritic cells in the blood. Yeah.
I think that what has been reported and we've observed by several studies is that there's an inverse relationship between the plasmacytoid dendritic cell frequency in the blood and viral load. I think that we've seen, everyone has seen, to my knowledge. What has become less clear is cause and consequence, meaning, as you mentioned, is it that because of viremia they redistribute so they haven't actually decreased, or is it that because of their function in greater access or crosstalk that you see less virus? And that's where I think the intervention with interferon might be useful, because you would say, if it's due to a functionality that you can perhaps exploit through interferon, then you should see some degree of clinical suppression, and we do. So because of that, I'm more inclined to interpret that the plasmacytoid dendritic cell frequency is an indicator of potential for control, but obviously it's in the circulation, so it's not really a direct effector site. So I just think it's a sentinel.
Dr. Prasad?
So, Louis, one question. So to follow up on Tom's question, so recent studies looking at latency reversing agents and what's the best marker to use or the best assay to use to detect cellular RNA expression, right, is showing that really if you just do RNA quantification, that there really needs to be distinction between the polyadenylated RNA versus total cellular RNA, because some of those transcripts should just be read-through transcripts rather than LTR-driven HIV transcription events. So do you know in Steve Deeks' study which transcripts were measured and whether really in the gut what's happening, there's just a lot more gene expression, so HIV RNA expression may be higher, but it doesn't mean that that would lead to virus particle production?
I'm pretty sure that they did address that in interpreting, but I'm going to go back and confirm. But I'm pretty sure they did. But I mean, I take your point 100%. I mean, I'm not trying to conclude at this point that that is accurate, but I am intrigued by the fact that all these studies have presented such a readable accessibility to the signal in the rectal tissue that it would seem to me that it is not a limiting amount of RNA. And if that is true, then that is consistent with our ability to detect it. But you know, I'm all for suspending interpretation until more data is available. But I do think that our focus on the blood is perhaps overemphasized, and that much I would say with confidence.
Reuben has been in the APOBEC business essentially from the beginning. He's made a lot of contributions, and I think he brings a lot to the table because he sort of looks at he takes a broader look at the problem, and I think he tries to consider some of the medical implications of all this, even extending to things like human cancer. So he asked me to keep it short, so I'll go ahead and turn it over to him.
Excellent. Thanks, Tom. Thanks, Rich, for the invitation, and all of you, congratulations for hanging on this long. That's a feat. So I wanted to keep it kind of later for you guys. I'm not going to give you a big data parade. There's lots of published pieces, but I do want to try to, I think the take-home message is there's different ways of thinking of the apobex, and there's at least 2 different therapeutic strategies that one could pursue. We're heavily invested in one of them because an academic lab can only, span so much bandwidth, but the other one is certainly valid too. This pointer is funky. All right, I'm just going to do that. So, does it help or hurt? And you could say that for the host as well as the virus. So the apobex, there's not just one of them. There's actually, in humans, there's 7 ApoBec3 genes, and they come in either single domain deaminase form, like ApoBec3A, C, or H, or double domain form, like B, D, F, and G. There's also 2 other family members, AID and ApoBec1, and this one, of course, is important for antibody diversification, and the other one is still debatable, but it definitely edits a couple of RNAs. So the ApoBec3s, and in fact, all of these family members that I portrayed up here have DNA deaminase activity. So they will convert DNA cytosines to uracils. And actually on the train this morning, there was somebody playing on a fancy phone, and they were doing some course online, and she was reading her questions out loud, and one of them was, what's the RNA base that is mutagenic in DNA? And I wanted to answer for her, but it was her. So it was pretty funny. But all of them catalyze this reaction, and when you convert the DNA-based cytosine to uracil, it's of course base pairing like thymine, and when that's replicated, it'll template the insertion of adenine, and that's mutagenic. So the apobecs, they're single or double domain deaminases, as I mentioned, and so a single domain looks like this, where there's a hydrophobic beta core, and there's one single deaminase motif per domain, and that's the active site, just to give you kind of a view of what they look like. So historically, this story actually emerged from a story where investigators, way before I even knew about HIV, were trying to grow the virus on different cell lines, and HIV would grow on, for instance, two different cell lines, two different T cell lines here. And when you take certain genes away, like in this case VIF, take the virion infectivity factor, small accessory protein, away from the virus, it's no longer able to grow on a cell that the wild-type virus was previously able to replicate on. And so Malam's lab Michael Malam's lab built on that real dichotomy to clone through a cDNA subtractive hybridization cDNA called ApoBec3G. And that alone could take when you overexpressed it, could convert a permissive cell into a non-permissive phenotype for the virus replication. So at that same time, and totally independently, I had cloned ApoBec3G and demonstrated that it had DNA mutating activity, as did AID and ApoBec1 and other family members, really suggesting that we've got a family of mutators. So that suggested a hypothesis for how this protein might actually be inhibiting virus replication, and that's depicted here in simple cartoon format. So one can produce viruses on a virus-producing cell, like 293 or HeLa. One can use a GFP-marked virus, so you have a marker for flow cytometry or visually through fluorescent microscopy for transfection of this producer cell. And one can harvest viruses, and you can play this game in the presence and absence of ApoBec3G and presence and absence of IF, and ask what happens to the viruses that are produced. And the prediction is, if it is antiviral, you'll get less infectivity. But the big prediction, if it's working as a DNA deaminase, is that this enzyme will convert cytosines to uracils in the cDNA strand, and those will template the insertions of A's on the plus strand, and that'll result in G to A mutations, which many others prior to us and Malam's study had documented in patient-derived sequences. So there's two predictions. You get less fluorescent target cells and a lot of mutations. And so this is one of those kind of eureka experiments where the first time it's like, whoa, there's a lot of fluorescence over here in the control without ApoBec3G, and here very few fluorescent cells. There's lots of potential explanations for that, but once you amplify and sequence some of the cDNA that was produced in that reaction, there's really only one explanation, and that is ApoBec3G completely mutated the viruses in one single round of replication. So this is just a small stretch of the GFP gene. It's just 600 or so nucleotides. But you can see every one of these sequences has a little, multiple, and in some instances, 20, 30, 40 little ticks. And each of those ticks are G to A mutations. So this is a very potent viral cDNA mutating enzyme. I'm distilling a little bit of older work to get to a bigger point here. So ApoBec3G, it prefers cytosines preceded by another cytosine. And on the virus's plus strand, that means it's converting G to A's in a GG motif. So this G converts to A. So you have GG to AG mutations. And you can see there's tons of them. You can stack them all up in lots of different formats. You can also distill it to a letter format where here's the base that changes, and here's the base next to it, and it predominantly prefers that motif. But then there was a little, everything's not quite explained moment when we compared this motif with what is actually observed in a number of clinical studies and reported in patient sequences. And there's clearly another component to that mutational mechanism, and there are other explanations for it. At the time, we were entertaining a few, but the most obvious is that one of those other ApoBecs must be doing the job. So for the better part of last decade, at least one person in my project has been chipping away at asking which ApoBecs are relevant to virus restriction. And to distill that to a model, we and others are close to agreeing on a model in which one would say there's a swarm of apobecs that have the capacity to do what I just showed you for apobec 3G. And so all of these apobecs, 3D, F, G, and H, can enter virus particles, travel with those particles until a new target cell is found. And then for apobec 3G, deaminate in a CC motif, and for the other ones, the TC motif, and literally destroy that viral cDNA. So this is awesome, but the virus has, as I mentioned before, that accessory protein called VIF. And so I'll blow it up on the next slide, but VIF works by promoting it binds, it recruits essentially other cellular proteins to form an E3 ubiquitin ligase complex that binds and degrades the apobecs. So the virus is smart enough to flush them out of the cytoplasm of a virus-producing cell before it produces oodles and oodles of virus. So that raises some therapeutic questions. Would one want to inhibit VIF? This is the obvious strategy. Let's stop VIF so the apobecs can really kick the virus's butt, right? But what I want to suggest is another therapeutic strategy, and I think both should be equally considered at this point. So what does VIF do? So this is a little higher resolution, still cartoon format picture, but we just, another lab just reported a structure, so we actually know how these proteins look now. But VIF nucleates a formation of a really interesting complex. First, it steals a transcription cofactor called CBF-beta, and then that heterodimer nucleates the formation of a larger complex. So VIF binds elongin C, Cul5, and those proteins bind other proteins. And then you've got an active E3 ubiquitin ligase, which can transfer ubiquitin groups to target apobec proteins, and that promotes their degradation by the proteasome. Classical mechanism. So, but what I want to point out here is that VIF makes 4 heterologous protein contacts, and all of these are potentially targetable. So one contact is the apobecs themselves. Another is a very large contact with CBF-beta. There's more than 5,000 square angstroms of buried surface there. And then there's a very conserved interaction with elongin C and Cul5. So I'd like to suggest that all of those are valid targets to go in there and potentially disrupt those contacts and unleash the apobecs on the virus. So that leads to this model. So if you remember anything at all from this talk, you just need to remember the word apobec and this bell-shaped curve, okay? And all the rest you can discuss over happy hour. So this curve is essentially a simplification of reality, but it communicates some points. So if you look at virus pathogenesis on the y-axis and virus mutation rate on the x-axis, HIV and most other species have come to terms with their mutation rates such that they're operating at an optimum, which promotes the fitness of the virus, but not, it's not so high that it's detrimental, and it's not so low that it can't evolve. And we know that one of HIV's really things that it's really super good at is kind of evolving fast, right, and changing fast. And we know that no two viruses are alike. We're fooling ourselves if we think they are. So when you think about it that way, it's possible that the apobec 3s are contributing to that optimal mutation rate that the virus enjoys and uses to beat antiretroviral drugs and T-cell responses and B-cell responses. And that VIF is more of a regulator of apobec mutagenesis and not so much like a light switch where it's off or on. And so there's 2 ways to think about therapy then. One is therapy by hypermutation. So if you inhibit VIF, the apobecs can destroy the virus, and I think that's a viable approach given all of our cell-based data. But the other therapy is a little bit less intuitive, and that's inhibit the apobecs themselves. So if you buy into the idea that VIF's just a regulator, let's add an inhibitor of the apobecs themselves to really stop viral mutagenesis from getting to this point, and then maybe that virus would be more susceptible to normal immune clearance, right? We just saw today several examples of suppression to varying degrees, and that's promising. It means, it may be controllable, right? So I want to just, for purposes of discussion, list things that support each of these ideas, okay? And then maybe open it up. So in favor of a hypermutation mechanism, so as I showed you, editing can result in lethal mutagenesis. So if you take any one of those sequences, they have so many mutations, including stop codons, there's no way anything functional is coming out of that. They satisfy something that I call Coffin's razor, which is the apobecs are just like antiviral drugs in that they can select resistance. So if you take VIF out of the system and ask the virus to grow in the presence of apobex, something will eventually emerge. Okay? So that says that they are a big enough threat that the virus has to evolve other changes to get around them. Okay? And they, of course, have naturally VIF. So this ligase pathway, as I said, has many potential druggable surfaces. So I focused on the 4 heterologous surfaces of VIF protein itself, but you can imagine 1 or 2 steps away also hitting a cellular target that would essentially disable VIF function. So the thing that's very important here is the apobex. So we just heard a very nice talk by Luis, which mentioned innate immune proteins. A lot of the apobex are interferon inducible, and that's dependent on cell type and so on. But one thing that doesn't really get appreciated by the field, and that's partly our fault, is that they themselves all exert a different selective pressure on the virus. So even though they all sound like apobex and they all deaminate DNA, they have different forces of power that the virus sees differently, and we're trying to tease that apart now. But you can actually liken them as essentially combinatorial therapy, using different mechanisms. So that's important. So a single drug could release multiple apobex. So, and then there's a potential for eradication if you buy into the idea, and nobody really knows where the latent cells are. There's some ideas and there are models, but I don't think we really know. So, but the apobex are expressed pretty broadly. So if the apobex are in those cells that are latent, and it can be combined with some sort of other molecule to bump the virus out of the latent cells, it's possible you could have lethal restriction at that point. So I would argue that this angle could be explored as a potentially, for sure, controlling therapy, but maybe even curative therapy. So there's precedent already. So Tarek Reyna and Mario Stevenson's group, and this is maybe a segue a little bit to the next talk. So of course, people have screened for things that inhibit VIF, and you come up with molecules like this one depicted here called RN18, where you add increasing amounts to a non-permissive cell and you get less and less virus replication. This looked pretty good initially, but it's a pretty toxic molecule and it hasn't reproduced in many other labs. But I think the concept of inhibiting VIF is still a very good concept and one that should be explored with a lot more, I think, energy and investment. The other hypothesis, hypomutation, and I saw a few of you go, this guy's kind of nuts. Maybe. So, but hypomutation is a good idea. So if we can turn down the level of mutational pressure, then we do have that adaptive, very good adaptive immune responses. So, and we heard a pulse strategy too, which is kind of clever. So if you can prime that adaptive immune response in addition, that might be pretty powerful. So the G to A mutations, as I said, are observed all the time in patient-derived sequences. And this tells us that VIF is not an on-off light switch. It's apobex are sneaking by a VIF all the time in vivo. Partial function VIF alleles also exist. So there's several papers out there, first Viviana Simon and Paul Benash, but you can imagine there's lots of VIF variants, and some of them aren't as good at neutralizing apobex. So again, you have more mutagenesis happening to the virus. So editing can also be sublethal. So we played some games with Lumansky where we grew viruses in the presence of apobex, and then we recovered them after that one round of essentially mutagenesis and showed that some can escape. So it's not totally lethal in one round. It can be, but you can also find things that sneak by. So G to A changes also result in drug resistance and immune escape, and that's easy to imagine. Anything that increases mutation levels will contribute to those things. These may be great targets to inhibit. These are non-essential enzymes, we would argue. So the mouse has one single apobex3. You can delete it, and it's aphenotypic until you challenge that animal with mouse viruses. We also lack one of the apobex3s due to natural deletion polymorphism, and some people are even unlucky enough to have hypomorphs of the apobex3H, which is the blue one on the right end of the locus. So a lot of people, probably even in this room, are actually shortchanged in terms of apobex also. So they've got 5 out of 7 instead of 7 out of 7. So I would argue that they're only serving as innate immune, antiviral, and antitransposon sentinels, and that they could be dispensable for short-term type therapy, like inhibiting the enzyme type therapy. And so again, as I've said a couple times, I think this strategy is also viable for suppression, control, and potentially eradication, because you would limit the size. If you did have very nice inhibitors of the apobecs, you could limit the size of the viral pool, the mutant pool within an individual, and maybe that's controllable by the normal immune response. So obviously we need to test that. So I'm not going to tell you about all our high-throughput screening, but over the last 5 years, in collaboration with Dan Harkai, who's a medicinal chemist at Minnesota, we've done screens now of over 500,000 compounds, and we have hundreds of candidate compounds like this one here that, this is just a cartoon, but it's the little shield that we can't quite see here surrounding the cytosine base that prevents APOBEC from knocking off that amine group. So we have real chemical inhibitors, and we have first-in-class and lots of other classes, and so we know these enzymes can be drugged, and we're pursuing this angle. So what I want to leave you with is this vision where, maybe APOBEC is an example of one of these base pairs to bedside, where, it was pulled out in a cDNA subtraction, and I cloned it by essentially some molecular games. But it comes from DNA sequence to function and a lot of insights in terms of mechanism and structure. And now we're at the point where we can realistically start thinking about ways of leveraging the APOBEC response, either by inhibiting it or unleashing it completely to control HIV. So thank you, guys. Time-wise?
I think you started 5 minutes late, and you nailed it, and we'll have questions at the end. Is that okay? So I'm not sure if this is a coincidence, but I think it's perfect that Rich is the last speaker.
There are no coincidences.
Because I think we should all together acknowledge the fantastic job he did in putting this meeting together. I can't remember a meeting that I've been to, and I go to too many meetings, where you just had all of the key players giving fantastic talks relevant to a specific topic. So it's just perfect, and to get everyone here on one day, somehow to get all the schedules to match up on one day is also a bit of a miracle. So I could go on and on for a while, but I won't. But I just think Rich has done a fantastic job, and I think it just is demonstrative of the good things that are going to be happening for HIV/AIDS research in Chicago as we're building and moving towards trying to help develop a cure. Rich.
Thank you. Well, thanks a lot. I'll try not to blush too much. And I have to say the hardest thing in the world is getting a lot of you guys in one room on the same day. So my topic is really sort of perfectly set up by Reuben's talk. And he's told you about the benefits of both sides of the equation, but I'm going to focus mostly on increasing APOBEC activity, because what I want to do is build on this restorative window of early HIV infection where the body can still fight back, and I think preserving APOBEC may be a key part of that. So APOBEC is one of the intrinsic immune factors, otherwise known as host restriction factors. And the hypothesis I have is that intrinsic immunity is maybe not the only thing, but one of the key things that the virus somehow destroys in the first 4 to 6 months after it first gets into a human. And APOBEC is an interferon-stimulated gene. It gets more complicated than that. But so are some of the others that you've heard mentioned today, TRIM5-alpha. There's also SAMHD1 and tetherin. But the focus today is on APOBEC. Honestly, I think for something really robust to happen to preserve defenses, we're probably going to need 2 or 3 of these. It's probably not going to be enough to have just one, but we got to start somewhere, and we're starting with APOBECs. And I'm going to tell you a story that has been evolving in my lab since the late 2000s. And the first thing that got us interested in having enhanced APOBEC activity, making more hypermutations, was the fact that we saw higher levels of APOBEC 3G in the, no, I guess I can't, in the physiological range could better inhibit VIF-positive viruses. So this is already a point Reuben has made. VIF is not perfect. It's not an on-off switch. And this is a different view than was common at the time that we did these experiments. At the time that we did those experiments, the view was that the amount of A3G left after VIF acted was just not enough to inhibit HIV. It was only enough to give HIV an advantage of being able to generate escape mutants from drug resistance pressure or immune selection pressure. But what we found was when we took Th0 cells and differentiated them in vitro into Th1 or Th2 cells, Th1 cells had 2 to 3-fold higher A3G levels, and that decreased the infectivity of the VIF-positive viruses they produced. And we saw a bigger effect when we used VIF-negative viruses. They were more inhibited, but there was an effect even with VIF-positive viruses. So then we said, well, we had a neighbor at the time who had a lot of elite controller specimens in his freezer. So we were interested in seeing whether elite controllers might actually, maybe they had more ApoBec3 activity. Maybe that was one of the things that helped them become elite controllers. So we looked at a bunch of B57 or B27 positive viremic controllers. These are people who had viral loads that were in the low thousands, around 5,000 for a long time, stable CD4 count, never treated with antiretrovirals. And we did sequencing of their proviruses, and what we found was that the A3G hypermutation index, this is a quantitative measure of how many mutants were in a stretch of DNA that we sequenced, that this was inversely associated with plasma HIV-1 RNA levels. And we also, this is a small number here, comparing the viremic controllers, also called long-term non-progressors, to non-controllers who were untreated. But we also found that plasma HIV RNA levels were inversely associated with the level of A3G RNA in the PBMCs. So this wasn't really conclusive because it was possible here. We were comparing untreated patients, elite controllers versus non-controllers. And the non-controllers had higher viral load. They had more HIV replication. That meant their cells had more VIF. So maybe they had less A3G just because their cells were expressing more VIF. And so it may not have been the fact that A3G was actually causing decreasing viremia. It may have been the other way around. So we did another experiment where we looked at resting memory T cells, not just all peripheral blood mononuclear cells, but the cells that harbor HIV's latent reservoir. And we compared them from viremic controllers, actually the same group of subjects, and this time a different group of ART-suppressed non-controllers. In this case, the viremic controllers actually had a little bit higher viral load than did the ART-suppressed non-controllers, who were all less than 50 copies per mil. And here we saw that the resting memory cells with the, in the viremic controllers, whether we looked at central memory or effector memory cells, they had higher levels of A3G protein than what we saw in the antiretroviral suppressed non-controllers. And the resting memory cells with the highest A3G had the lowest levels of provirus by ALU-PCR. And VIF-positive virions that we reactivated in the lab from cells that had higher A3G had lower infectivity. So this was endogenous virus being reactivated in the lab that was VIF-positive, but it wasn't able to get rid of all the A3G, and if you had more A3G, the virus that came out was less infectious. So the conclusion from this study was that controllers have 2 to 3-fold higher A3G during chronic infection than non-controllers. All of these subjects had been infected for years, okay? These are all very chronically infected subjects. And we couldn't explain the fact that the controllers had higher A3G by the fact that there was less VIF, because in this case, they actually had higher viral load than the comparison group, which meant maybe they had higher VIF levels. So what we wonder is whether, and this is still a theory that's out there that we haven't explored more, but we wonder whether the controller's early CTL activity might have better preserved their A3 intrinsic immunity. Because we know early in infection, these controllers do have good CTL activity. Eventually, the virus escapes from that. So maybe there are multiple mechanisms that allow these controllers to have persistence of their control even after their CTL changes. And other evidence is consistent with the idea that A3G may decrease after acute infection. So there are, let's see, how many groups? 1, 2, 3 groups that have independently reported no evidence of A3G hypermutation in plasma virus RNA from chronically infected subjects. Our group was one of them. But I was shocked when I read a paper from Beatrice Hahn's group, and then a subsequent one came out. Beatrice looked at clade B virus, a subsequent one looked at clade C virus, and when they looked at viruses from the very initiation of infection, what are called transmitted founder plasma viruses, in the plasma of these acutely infected subjects, they found tons of A3G hypermutation. And we know from the work of Viviana Simon's group and others that VIF does adapt over time to better degrade a stable haplotype of A3H. I think Reuben has some similar evidence. And we also know that VIF has adapted to better degrade apobex, and that has allowed cross-species transmission of SIV sooty mangabey to rhesus macaques. So the hypothesis is that some cellular mechanism may down-modulate A3G as one of the interferon-stimulated genes soon after acute infection. And we really don't have any longitudinal data about whether this happens. So I was very pleased to recently receive a grant from AMFAR to try to get some data like that. So we're going to be doing an observational study. The IRB just approved the protocol 2 days ago. And this is going to look at untreated, very early infection. This study is taking advantage of the U01 cohorts that Brian Mustansky described to you this morning. And we're going to test the hypothesis that the expression and function of A3G decreases over time during early untreated infection, and also look at other ISGs to see if any of these other restriction factors may also change over that time frame. And what we're thinking about is that this happens in that first 4 to 6 months. That's the restorative window. We think this is when all these events might be happening. So we're going to compare 2 time points looking at hypermutation in plasma HIV RNA, resting memory cell A3G RNA and protein, and we'll be looking at other ISGs and restriction factors too. And this is all possible because in the cohorts that Brian and his colleagues in the IMPACT program have established, they're doing 4th generation HIV testing 2 to 3 times a year in these high-risk young gay men, and they have a 2 to 5% annual HIV incidence in these cohorts. So we'll let you know in a year or so what we find. Now, the benefits of focusing on early infection and trying to do something to preserve those defenses that might be lost by 6 months are that we might limit spread as well as benefiting the infected persons. They may have better long-term outcome of ART, even if they only have a brief remission. Early ART may limit the inflammation. It may be that the inflammation that causes a lot of the premature aging that's a big problem now is established very early when interferon responses sort of self-perpetuate. And if we could do something to better knock out the virus, maybe that would decrease that self-perpetuating interferon response. And I suspect that something in addition to ART is going to be needed to increase the percentage of people who get to a sustained remission after very early ART that you heard about from the Visconti trial or like the Mississippi baby. So the idea is maybe boosting cellular factors up to and including integration, ApoBec3s and cellular DNTP synthesis. That might be a way we could add effects on the host cell to our current antiretroviral agents, and if we give them early during acute infection, early infection, maybe we'll have more benefit. So we've started taking some next steps. We're looking both at APOBEC and at DNTP synthesis, but I'm just going to tell you the APOBEC story today. And the idea is that if we do see that APOBECs decrease, we're going to need a novel intervention that will boost A3G. And this would be for a proof of concept trial where we take a few patients and add this A3G booster to antiretroviral therapy, and then after a year or two of therapy, do an analytical treatment interruption and see if we get a higher proportion to have a sustained remission if their APOBECs have been boosted. So we have taken the approach that's very similar to what Reuben has told you about, except we started by trying to use RN18 and another compound that was described in the literature as controls for high-throughput screening and found they didn't really work in our hands. So we set about trying to make something that we could use as a control for high-throughput screening, and we have cell lines that stably express A3G luciferase fusion protein to identify small molecules that might boost expression. So this shows that luciferase expression, that's just relative light units, is driven from a CMV promoter, and clone 10 does not express luciferase, that's our negative control, and clone 5 expresses more than clone 6, and this is very stable. And A3G luciferase actually gets into VIF null virus and can restrict in TZM-BL cells. So the tag on the end of the luciferase tag on the end of A3G really doesn't seem to impair its antiviral function. And these A3G luciferase cell lines are sensitive to VIF. When we transfect VIF into these cell lines, we see degradation of the A3G luciferase fusion proteins, and this happens for all of them. And when we infect with VIF-positive NL43, we can see that there is some decrease in the RLUs, that VIF is in fact degrading the APOBEC in these cells. And with the help of Gary Schiltz, a chemist at Northwestern in Evanston, he did some chemical modifications and made compounds that we screened, and we found that NU52 was effective at boosting. This is what happens when we use 15 micromolar of NU52 on either the luciferase number 6 or number 5 cell lines. If we just add DMSO, the vehicle control, you see no increase in luciferase expression, but you get a 1.5 to 2-fold increase in expression at 48 hours when you add the NU52. And we compared this to the other compounds, RN18, and the other one that's described in the literature is in green, that's IMB16. And unfortunately, IMB16 was pretty toxic to cells and decreased their proliferation. And really, neither of those compounds did anything much to APOBEC expression in our system using these A3G luciferase constructs. You can see that here, that if anything, with increasing drug concentration, as the cells were dying, we see less APOBEC expression. But with NU52, we don't have any problems with cell proliferation, and we do see increases in APOBEC expression. And NU52 also increases A3G and A3F protein levels when we look at native protein, not just the luciferase fusion. This is using Western blotting to look at either A3G here with NU52, we see an increase. And this is a mutant of A3G that is poorly expressed, and we can salvage that expression with NU52. And we also see increases in A3F, another important mutator. And we don't know for sure, but this data makes us wonder whether this compound may work by decreasing degradation of A3G rather than increasing its synthesis. But in either case, we can show that NU52 enhances the activity of A3F and A3G against VIF-positive viruses. In this experiment, 293 T cells were pretreated with either control, that's DMSO in the darker shade, or NU52 in the lighter shade. And this just shows a cell that was transfected with a control vector, but if we transfect the cell with a vector expressing A3F or A3G with DMSO, we don't see this is sort of the baseline level of suppression of virus replication with DMSO, and we improve virus replication when NU52 in the lighter shade is applied. And this is taking the viruses produced by the cells that were treated with NU52 and then measuring their infectivity on an indicator cell line. And so there is less infectious virus produced from cells treated with this compound. And recent data suggests that waiting 3 days before we start harvesting virus is better than 2 days. So the 3-day treatment increases A3G levels up to 3-fold. This is still just a tool compound. It's just a start. It only has an IC50 of 3.2 micromolar. That's not enough to think that we're going to be able to move this into something we can use in people, but it gives us a compound we can use as a control for high-throughput screening, which we're about to start. And it also gives us a compound that our chemist colleagues can continue to manipulate. They tell us there's lots of room for further modification. So to summarize, A3G luciferase stable cell lines expressing different levels of A3G and also co-expressing HIV VIF can be used for screening to find boosters of A3G. And these proteins still restrict HIV, and they are still sensitive to degradation by VIF. And we have a tool compound, NU52, that enhances ApoBec3G expression and does not decrease proliferation of activated CD4 cells. And it also enhances antiretroviral activity of A3G against VIF-positive wild-type HIV. And we see that the earlier compounds that were described by others, as Reuben has already alluded to, don't seem to really work. So I want to thank the people who really did this work. In addition to Gary Schultz, who's at the ChemCorps at Northwestern, Chisu Song did almost all of the work that I showed you today with a lot of help from Harry Taylor. Some of the earlier work was contributed to by Michael Vetter, John Donahue, and also Maria Pia de Pasquale, who I forgot to put on the slide. And I'm really excited about working with Brian's team and the IMPACT program on that new AMFAR study that I told you about. And also very grateful to be participating with Brian's U01, which is funded by NIDA. Thank you very much.
So Rich was perfectly on time. And so now...
That's an accident.
So I guess I'll ask the first question, which is one of the confounders for VIF-targeted drugs is the idea that, ApoBec-targeted drugs is that if you influence proteasome function, you could be influencing the turnover of it. So do you have what's your plan to try to address that?
Well, we actually have been exploring whether proteasome inhibitors might lead to boosting ApoBec, and so far we've drawn a blank, but we're still looking at that. I don't know if you want to add anything to that.
No. The proteasome inhibitors will rescue some of the VIF effect
Yes.
on ApoBec.
Yes,
but
that's
they
right.
won't
But they won't change
ApoBec itself in a non-VIF expressing cell.
Right. Yeah, it's not really clear what pathway the cell uses to degrade ApoBec in the absence of VIF.
So Harry, and since you work for Rich, I hope you're asking Reuben a question.
Of course. So I'm really curious about your opinion as to the contribution of the A3Gs and producer versus target cell effects. And are there any systems that you've been aware of that have analyzed the potential interaction with those different potential mechanisms? So if you have both A3G on both the producer and target cell side,
Yeah.
are they additive? Is there some
no,
synergy?
No. So it's just a producer cell effect. So the APOBEC, for it to be effective, has to get inside the virus's capsid. It's capsid-enclosed core, where the nucleic acid is reverse transcribing. And that core, there's evidence from the trim area, and there's beautiful conical things being produced by the structural biologists and stuff. But that core, the way I view it, is semi-permeable, so little things like DNTPs can get in, but big honking things like proteins can't. So that core protects the nucleic acid until quite a late stage of reverse transcription, when it's probably almost done and then integrating. So target cell APOBEC has no role whatsoever.
So you don't believe any of the A3A studies in myeloid-derived cells?
Well, believe's a strong word. I never use in science. So
Save it for Sunday.
yeah.
So you don't believe, so the data is not consistent with your observations?
So we haven't explored myeloid cells. We've done a lot of work with T cells and other cell types, and we see no target cell effect. And APOBEC3 is not also in a T cell. So
Rich is
So
going to chime
I'll
in.
have to respectfully disagree and refer you to a couple papers.
no, I've seen a couple papers.
And it's not the effect that was originally claimed to exist. It's a small effect, but I think there's a possibility that there is a target cell effect even in T cells from A3G and A3F.
I don't think so.
I'm with Reuben.
And we've done, I didn't
Well,
kill you guys
you've
with
read
data.
our paper, so
Yeah.
tell us.
So we've
the
done a
criticism.
lot of experiments with endogenous APOBECs. So we went in there and we've deleted APOBECs and we've systematically
Yeah.
knocked
Oh,
them
yeah.
down, and we get no difference in infectivity of those cells. So, that's the best, in my mind, that's a gold standard experiment when you're manipulating the endogenous APOBECs in a very clear, especially the deletion experiment, uncut way. So.
I had two questions. So first, have you considered that the effect that APOBEC that you can see at the beginning of the infection is due to the small population size and not that APOBEC activity decreases through time? So what we have seen is sometimes when the population is heterogeneous, you don't see so many hypermutants. But when you have a very small population, because they cannot be deleted from the population or they cannot be eliminated, you start to see these hypermutants. So when you have the initiation of the infection, the population is very small, and then you can see those hypermutants.
Well, I think that's a valid point, except that in the papers that they were looking at plasma RNA levels that were in the millions. So it may be a homogeneous population, but there's a lot of virus there.
Yeah, but the initial population is very homogeneous. It's not the number of viruses, but the number of mutants that. As long as the population generates more heterogeneity, it's more robust, so it can eliminate those deleterious mutations or hypermutants. And the second question was to use APOBEC with RT won't be an opposing evolutionary forces. So one is diminishing the heterogeneity or the replication, so then the heterogeneity, and the other is increasing the heterogeneity, so it will be a little bit conflicting.
So I would say they're in terms of genetic heterogeneity, they're both contributing factors to the overall diversity of virus that's observed. So we don't know what those relative levels are, but they're both clearly big factors. So I think at low levels it's contributing, at higher levels it's diminishing. So it's a threshold. Yeah.
The other point I would make is more of a clinical point. We know the signature nucleotides where APOBEC is going to cause mutations. So that means we can identify which drugs are prone to escape, their inhibitory. The virus can escape their inhibitory activity by making those mutations. So we'd have to be careful to construct antiretroviral therapy regimens that didn't include drugs that could escape via APOBEC-mediated mutations.
Yeah, that's a good point.
Yeah, can you comment on like the interferon resistance observation and acute infection? Because if I'm hearing all the information correctly, you're saying it matters to have APOBEC in the particle, but you have little hypermutation in end-stage disease to suggest that there's less impact of APOBEC at the time of transmission, but meaning in the particle itself. But then once you get into an acute infection setting where you do have interferon pressure again, is it the VIF that has not necessarily been called into action that all of a sudden is called into action in acute infection, or you think the observation from the in vitro replication has nothing to do with APOBEC relative to the interferon resistance?
Well, I think in acute infection, the interferon response may be causing higher levels of APOBEC in some cells, at least. And maybe that's an explanation for why you see more A3G hypermutation in viruses from acute infection than more chronic infection.
Because Beatrice is interpreting that she's cloned the sequence of the founder, and because it has less replication by P24 in vitro, is interpreting that that means that the transmission is favored for the interferon resistance profile. So I'm trying to reconcile the way you view the world.
I can't reconcile it. I have the same sort of confusion you do. I don't really get it.
Those are very, I mean, if you consider Beatrice's data, and she's looking out over 10 days, and she sees a 10-fold difference, that's a very small difference in each round of replication.
I agree. I just
So
didn't know how much it should be weighed relative to what you're presenting. I'm basically saying it shouldn't. That's fine.
Yeah.
So I have a question to Reuben.
Sure.
So it, actually two questions, and it grow three while I'm waiting. So the first one is in the human APOBEC, they have a 7 APOBEC3 protein. So what's the opinion why human has the most of them among all the APOBEC family? That's my first one.
So let's do one at a time then, or I won't remember. So all primates, all higher primates have 7 that we're aware of. So there's variability within the lineages. If you go into some other mammalian lineages, like for some reason, ancient selective history probably, horses, for instance, have a lot of APOBECs, whereas their, related farm animals have only 1 or 2. So I think it's just, for whatever reason in the primate evolutionary history, there were selective pressures that favored the expansion of the locus. It's consistent with its overall role in innate immune response to viruses and transposons.
So my second question is, you propose it to inhibit function of APOBEC to approach how to tackle HIV prevention or cure. And then if you block the function of APOBEC, the other role of APOBEC is blocking the transposon and the endogenous retroviral movement inside a cell. So how are you going to address that one if you block the other function?
So on an evolutionary scale, I think that's a really important function. I think within an individual, it's probably dispensable for some length of time without any obvious phenotype. What I worry about a little bit more is susceptibility to other viruses that we don't know about. So there's APOBECs, the classic here is they're very good at stopping zoonotic transmissions. They're a very, very potent barrier for when a virus is trying to go from one species to another. So VIF is able to help it adapt to do that, and it has in the past. But I think when you take them all away, it'll be interesting to see what, we have to be careful.
So my final question is, while I'm waiting, you mentioned that you made a cell line that knock out the APOBEC gene, and then you already have a system to test whether your approach is going to be valid or not. So
no,
we
use
don't. That cell line and then test it whether it responds better to the drug?
So it's a good system for virus, essentially mutation studies and infectivity studies, but it's not a good system to test the hypomutation hypothesis because we actually need an animal with a fully functioning adaptive immune response. So for instance, maybe, something like the, if a monkey model, if the assuming that our inhibitors work as well against the monkey apobex as the human apobex, but we need a fully functional adaptive and innate response to test that hypothesis. Because the idea is that the adaptive response will be the controlling factor when the virus is less mutable. Yeah.
All right, great.
Thanks.
Still on time? Can you ask him at
Really quick? Oh, okay.
Go ahead.
So, the data, so are you in the camp that believes or supports the idea that Eplebac mediates all its activity via its enzymatic activity?
It depends on the target. So for HIV, the lion's share is deaminase dependent. For other substrates like line 1, the data are absolutely black and white that it's deaminase independent. So it just depends on substrate. Yeah. Thanks.
That last question drilled deep into the bowels of the field. Thank you very much for a fantastic series of talks.
Okay, I'm going to step back into the moderator role and apologize for the excess jargon in that last session, but you all get some sort of award for sitting through it and asking great questions. Reuben and I probably haven't had this much fun in a while. The final thing I asked for a conclusion to this conference is for a group of our local leaders, including some leaders of important community organizations fighting HIV in Chicago, to come up on stage and give us some comments about cure in general, what they may have learned from the conference today, and really in particular, I would ask a pointed question of our panel about how they think we could operationalize cure research in Chicago. So Brian Mustansky is here, so I'll ask you to come up, Brian. David Manar is, for 3 weeks now, been the director of the Howard Brown Health Center. And Tico Valle is not here yet. So he's the director of the Center on Halstead, and if he joins us, he will. Oh, good. So David, I'll let you start with your thoughts.
There it is, folks. Oh my God. Good afternoon. Thank you. Thanks everyone. Really fascinating forum. I learned I should have paid more attention during college, undergraduate biology. So you lost me there on a couple of the presentations. It's a fascinating moment in the epidemic. I guess I wanted to kind of go back to Timothy's presentation and kind of bring your story kind of back to round out the day. Fascinating turn of events that you lived through, and also that you have taken your experience, Timothy, to galvanize public awareness about research for a cure. You're called a Berlin patient. As Nick was presenting the epidemiology slides for Chicago, I'm thinking I'm one of the Chicago patients, diagnosed at 24, not unlike some of the young participants in Brian's study. And early in my diagnoses, praying, would I be one of these elite long-term controllers? Is this, knowing that this was, a rare group, but would that be possible? Would there be something early on that could stimulate my immune system that could save my life? In 1994, there was no effective treatment. And also fearful of, back then the experimental treatments were causing tremendous harm. And we really didn't understand how to mobilize ARVs and dose them in ways that could minimize side effects, until late 90s, early 2000s. So I just, kind of listening to Nick's presentation and your story, Timothy, really had a strong reaction about some of that. I think this is it seems like we're really on the beginning of this field. There's a lot to learn, Hopefully we'll learn about a functional cure or a sterilizing cure, or better information about immunology and virology that could have other unintended benefits for HIV treatment or other disease states. So that's very exciting. The public really doesn't understand this work at all. The public really thinks, and particularly law, I mean, most of my background was at the AIDS Foundation of Chicago for 20 plus years, and a lot of that time spent with lawmakers and policymakers and trying to raise awareness and government investments on this type of work. And they really think of the work as, do we have a cure or don't we? Is it here or don't we? And so I think, we're going to have to do a lot of education about this, kind of the incremental work around this. And even in HIV communities, I have a good friend from the UK, Gus Cranes, who's been doing a lot of education among people with HIV that today it's not just HIV positive status or HIV negative status. And he's been promoting this idea that there's actually 5 different types of HIV serostatus. HIV negative, not on treatment. HIV negative, HIV negative, period. HIV negative on PrEP, unknown serostatus, HIV positive uncontrolled, HIV positive virally suppressed. And hopefully one day we could add to that, HIV positive in a restorative window, or HIV positive in remission. But that's going to take a lot of education to kind of get folks to rally behind this. So it's hard work. I commend you all. And I also think we should really think carefully about what a cure means. What is that? What are the attributes of cure? As I said, I've been living with HIV for 20 years. Thankfully, I'm virally suppressed. For all intents and purposes, I feel like my HIV has been cured. What I've not been cured of has been the stigma, the fear that my treatment will fail, or that side effects will creep into my life. But thankfully, I'm living an experience that feels very close to a cure.
Thank you. Well, let me just begin by thanking Rich for organizing this. I think it is such a phenomenal opportunity to bring together people from the community, organizations, from the health department, from the biomedical sciences, the behavioral sciences, and I think that that's really what it's going to take for us to move forward with addressing the HIV epidemic, whether it be a cure or other approaches. And I think this is a rare opportunity, a real sort of gift that we've all been given to come together and learn from each other. And I think what David just said is really an insightful example of that. Talking about these different ways of thinking about what it means to have HIV, to be virally suppressed, to be someone on PrEP. And we'd heard from John already today that idea of talking about condomless sex instead of unprotected sex. And I think that's really an example of sort of how we need this interplay between the biological researchers and the behavioral scientists, the epidemiologists, because in the work that I do, this is a challenge to us to rethink the way we study HIV risk. We write in a lot of papers, engaging in HIV risk behaviors is a phrase that if you type into PubMed, you'll get thousands, if not millions, of citations. But what does it mean? What does HIV risk mean in the context of these new treatments, in the context of cure? And so I think that those are the kinds of answers that are only going to come from this dialogue between community members, biomedical scientists, behavioral scientists. So I think that's really exciting, and I think it also reflects this multi-level perspective that we really need. I think the other takeaway is, hearing the excitement around treatment, both as prevention, but also early treatment as a mechanism of cure. I think it presents opportunities for us to all rally around the idea of making treatment available to people, breaking down barriers for people to know their status and get access to treatment for now many potential reasons, in addition to their own health care, the likelihood of reduced transmission to others, the likelihood that they may be potentially a better candidate for cure down the road if they initiate treatment early. So I think that these kinds of the discussions we've been having about HIV cure can lead to us reframing the way we think about a lot of existing processes that exist around HIV. Testing, for example, we were talking over lunch, how much testing would change in this country and the world if part of it was to get tested so that you could get cured. I mean, that's a very different paradigm for talking about testing versus getting tested so that you will have a lifetime of use of some very effective medications. Those are different stories. And so I think that this dialogue around cure will reframe much of the way we think about HIV down the road and, you know, from testing to treatment. So I think it's very exciting, and I'm happy to be part of these discussions.
Well, I'd like to open up the floor. If anyone wants to make a comment or ask a question, feel free. I think it's incredible to me to hear different perspectives today that I've really not heard before. You know, I've been doing this for a little while. So, I mean, coming together with people from other disciplines, you know, this is a very validating experience that works. You know, what you just said, Brian, I think is potentially really important. If we can frame participation in, you know, your own healthcare and perhaps subsequently participation in research around making it more likely that down the road when a cure is available, it's going to work for you, that seems to me like a good win-win for science and for public health. Nanette, introduce yourself, please.
Nanette Benbow, Deputy Commissioner for STI/HIV, Chicago Department of Public Health. Sorry for the long title.
Thank you.
So thank you, Rich. I think I've been doing what I do for a very long time, and I welcome having a full day or 3/4 of a day of basic science, and have learned some. And I really appreciated even last night having a slightly different conversation. We have forums and meetings all the time, but yesterday's conversation, with the idea of cure and all the possibilities that brings, re-energizes the conversation. And I think it's just one other tool that we have in our different ways of trying to deal with, with the epidemic. And certainly from a public health perspective, we really appreciate that new perspective, which, is now catching on. So thank you for igniting that.
Well, thank you for that comment. Well, Debbie, please.
Actually, I wanted to ask David, thank you for sharing your story with us in terms of the perception of false optimism with the use of the words cure. And, we sort of have qualifiers in terms of functional cure and sterilizing cure. But from a patient's perspective, I mean, what do you think we should do as biomedical researchers and clinicians in terms of paving expectations towards cure? Because it's such a loaded term. And, for pediatrics, we've sort of used remission, but it still doesn't get to the meat of the matter in terms of the long road ahead for the world at large.
Thank you so much for that question. I've been thinking about this and trying to figure out how to frame that, this kind of this point. I think for patient populations, particularly people that have access to care or are on treatment already, obviously there's an exciting prospect where they could be defined as cured, and everybody can understand that intuitively. But I think understanding also the how the science is still complex, not completely understood, could have side effects or unintended consequences. I think we do have to kind of frame the value proposition. What is that value proposition? Is it, stopping, or not needing to rely on daily treatment? Is that the value proposition? Is the value proposition that you're guaranteed that your treatment will not fail? Is the value proposition that you are not going to develop neurological conditions because of HIV or advanced aging? Or inflammation? I mean, what is that value proposition? I think we have to frame that for patient populations. It's not entirely clear to me what that is right now, and except around stigma. So, the idea of being able to say, I don't have HIV is really powerful. And that's why I commend Timothy for telling your story. Because I can think of 100 people I know that would, if they had the opportunity to be cured, they would not want to be identified with HIV ever again. And they would run far away. So stigma is a big one. Stigma is a big issue. And stigma is actually one of our biggest challenges if we're going to try to reach people early in their infection or their diagnoses and try some of these interventions that could have lifelong benefits, but we need to make a big intervention early in their infection because stigma clouds people with so much shame and blame and uncertainty and depression and trauma. So I don't know if that answers your question, but I think it is an important one.
I would just add to that. I've been thinking throughout this discussion about what it will mean for people who don't benefit from the cure, if and when it would arrive. People who either because they were caught too late in their infection, or were just because of certain characteristics they had, it didn't work. And so do we then create sort of two classes of people who were cured and people who weren't cured? And I think that is a real big issue that we can't take lightly when we think about issues of stigma.
Luis?
I basically just want to get your feedback on the community's buy-in to cure-related study participation, because as new strategies get initiated or conceived, one of the first groups that everyone wants to start to work with are the individuals that have done very well on therapy and are very predictable and very stable and suppressed and so forth. And these are the same people that have gone on with life. And, HIV is not really what they're thinking about anymore. And one of the discussions is always, get your community groups, really informed about the research you're doing, get them involved in the process, and they will go out and advocate within their community for the studies to move forward. Another is, emphasize the altruistic issue that, the benefits that you're receiving now due to the therapies that you're taking were because somebody took a risk. And then you have to frame the benefit differential, saying, what would you gain if a cure were to be developed? And I think this issue is one that we wrestle with in the research side, saying how do we best engage? Is it the co-develop a process, but then you have to get buy-in from the very beginning, or is it that you have to frame the differential of the benefit and the community is already hungry for that, so they will deliver the recruitments? Because there's going to be more activity, I would expect, in the future, right? You have $100 million that are being committed to the cure research agenda. There are study concepts moving through multiple different platforms. So eventually there's going to be a conversation that's going to have to be made relative to is there enough interest in the cure agenda for people to sort of step back from the benefits of therapy and put themselves back into this quote-unquote research mentality.
Well, I think one thing that's probably common to both of us is we're both coming from this, both as HIV researchers, people who are very passionate about HIV, but also people who work on the LGBT health movement. And I think that is a different angle, a different perspective when you're coming at work on HIV, because I consider myself foremost an LGBT health researcher, and I think of myself as someone who's embedded in the community and is working on the health issues that are disproportionately experienced by the LGBT community. Obviously we looked at the epidemiological data earlier that 70% of infections are among gay and bisexual men. So HIV is a critical health issue amongst this community. And I think it changes the conversation to some extent when you're looking at it from the community health perspective versus looking at a particular disease and sort of trying to advance an agenda around a single disease. So I think there's a lot to be said about being sort of embedded in the community and sort of having this long track of focusing on community health of the most affected communities. I think it changes the perspective a little bit, and I think it means that we're sort of in constant dialogue with gay and bisexual men, leaders of the community, members of the community. You know, when you were talking about sort of working with participants who are already doing really well, I mean, I don't think that reflects the young men that are necessarily in my cohort studies who struggle to make their research appointments. And, you know, there's many who are doing great and many who are also struggling. And so I think because of our position, because of the way impact is structured, because we're embedded within a community organization, you know, we really do get a cross-section of young gay men to participate in our study. They're racially diverse, SES diverse.
I wanted to add to what Brian's saying, and I wish Sybil, who presented today, this morning on PrEP research with young gay men, principally gay men of color, were here because her research is really, for me, really demonstrative of the value proposition around research. So her study, which around PrEP for young gay men, occurred before PrEP was approved by the FDA, and subsequently after, you know, was targeting this group, mostly young gay men of color, and you know, and introducing PrEP to them and regular testing, and there was a behavioral intervention component and condom distribution and a lot of counseling about, you know, their sex lives and their concerns and their health, and some of the young men selected PrEP and were successful on it. Some of them didn't. Some of them weren't able to adhere. But unintended from the study, they found that the young participants of this research project really bonded with the clinical team because they had never had a primary care experience that actually embraced them as whole human beings with agency, with a right to a happy, fulfilling sex life, with, you know, challenges in their lives. They had this whole care team that was caring for them and listening to them and engaging them, and support groups and all of this infrastructure that they had never experienced. And so there was a lot of affinity to the research project because of that. And it was sort of a sad statement about what our, you know, our non-research settings provide or fail, or in many cases fail to provide vulnerable individuals. So I think we can make that value proposition, but it may not be about the altruism, and it may not be about the direct benefit. I mean, in part it could be, but it's also going to be about the other ways that we, you know, augment the service plan for individuals and really support them in all of the facets of their life.
So, David, that really resonates with me, and I think Dr. Fair and some of the other older folks in the audience may agree with me on this. In the early days of HIV research, before medicines were widely available, being a participant in a research study was something very different than what it is now. You know, it was, you were really gaining something special. You weren't sure that it was going to work, but it was trying to do something to save your life, or just sitting back and, you know, writing a will. And so to some extent, I think what we have to figure out is how to make the value proposition, if you participate in this research, we're going to help optimize social services, your immune system, you're going to have a limited amount of virus persisting in you. You know, we shouldn't be talking about the cure because we're not there yet. But we could be talking about ways in which we could improve the health and well-being of people by having them participate in research.
Mental health.
Mental health.
This is obviously an APOBEC effect because if the early cure attempts don't work, the deamination of the cure will turn into care. So.
Oh, I like that. APOBEC is essential for cure to turn to care. Well, I can't resist saying that's got to be the tagline for everything going forward. Well, I don't want to cut this conversation short because it's been a fabulous one, but I'll also say that waiting for us Well, in about 4 minutes outside is a very nice reception that you're all invited to with some food and drink. And so we can continue the conversation out there and enjoy a little food and drink. Thank you.